to continue our discussions in biochemistry lab. Again, as a recap, we have here the diffusion or the osmosis process or dialysis process, wherein water passes or permeates through a semi-permeable membrane. So usually, if you try to see, water can permeate both inside or outside environment of the cells. So we have metabolic toxins, urine and small proteins and blood streams usually crosses the cell membrane, and even water crosses the cell membrane, and then it is considered to be a reversible process wherein water can go back and forth across the cell membrane or crosses the cell membrane, I mean. Next, we have test for chloride. So 1 ml of the dialysate. Dialysate is the based on the dialysis setup. So we have the stock solutions, wherein stock solutions of chicken stock solution cubes are boiled and put in the gastric lining, and then the gastric lining are being tied up both ends, and then macerate it or leave or stand it for 1 hour in the distilled water or in the water. Then after that one, you have to collect the dialysate and add 1 ml silver nitrate. So the positive result is white precipitate, wherein it indicates that the stock solutions diffuses or crosses the gastric lining through its cell membrane, and then it contains a chloride, because stock solutions of chicken contains chloride or salts of chloride. Then if you try to add silver nitrate, then it will form a silver chloride, which is white curdy precipitate or white precipitate. Next, 1 ml dialysate, that's 2 ml sodium hydroxide plus 5 drops of 0.5% copper sulfate. Usually there is a violet color that is positive for peptides or proteins, wherein that stock solutions inside the gastric lining contains a chicken, and a chicken contains a protein. So positive siya. Okay? And 3 cc or 3 ml 1% albumin solutions plus ninhydrin. So you have yellow colored complex. Then it will form purple for the presence of amino acids. Okay? For diffusion process, based on that procedure, in the petri dish, you have hot and cold water. So you have to add pinch of potassium permanganate and a pinch of potassium permanganate in the cold water, and this is a warm water. So you try to see the diffusion here is faster compared with the cold water, because the principle states that in a warm water or boiling water, the space of molecules are large enough, while in the cold water, the space of the molecules are closer to each other, and there is erratic movement of the water molecules, which lead to the faster diffusion of the solutes, or represented by potassium permanganate. Okay, so principle involved: the higher the temperature, the faster it is diffusion process. Next, so if you try to see the diagram, we have the movement of molecules, net flow of molecules across the cell membrane. So it can flow back and forth as long it is the size is smaller, that can fit to the membrane layer. So the passive movement of molecules or particles along concentrating gradient, or from regions of higher to regions of lower concentrations, that is passive diffusion, usually from higher to lower concentrations. Next, passive transport or passive diffusion process is the movement of molecules and other atomic molecular substances across the cell membranes. If we try to see, this is a passive diffusion, simple passive diffusion. So molecules crosses the bilayer of phospholipids, and we have here another one, molecules passage through the protein structures or secondary transport or a carrier protein. Okay? Example, yung mga carrier protein, glucose, it needs glucose transporters. So facilitated diffusion process. So transport of substance across biological membrane from area of higher concentration to an area of lower concentration by means of carrier molecule. So since the substance move along the direction of the concentration gradient, energy is not needed because higher concentration here and we have a lower concentration inside the cells of the cytoplasm. So there is what we call a faster movement of solutes coming from higher concentration to lower concentrations. So insoluble molecules, so we have here carrier proteins, for example, the movement of the glucose, so it needs glucose transporters. Movement of other molecules, it needs a carrier, or even it is stimulated by a stimuli that binds with the receptor site, leads to the opening or the activation of the carrier. Okay, next, so we have active transport. It is the movement of substance against the concentration region from low to higher concentration in all cells. That is usually concerned with accumulation of molecules that the cells need, such as ions, glucose, amino acids. It is the process uses energy such as adenosine triphosphate inter or primary active transport. What do you mean by this one? Usually, the movement is from low concentration, from uh, low concentrations to higher concentrations. Usually, if they have a low concentration of the sodium or potassium inside, so it will goes